Hi again, welcome back. We are just starting our quest into mitosis and cell division, and later on we'll get into meiosis with gamete cell division. So we left off talking about the chromosomes and to give a little background on chromosome structures. And before we keep going down our list for definitions on chromosome structure, I want to give you a little bit more a visual before we do a couple more definitions. So far we've talked about what a chromosome is, how it's made of chromatin, and how the arms of the chromosomes are called chromatids. So let's define that a little bit more, and then we'll have a couple more definitions under big letter B to help you out. So I'm going to draw from almost from here on out in this section, I'm going to be drawing cells that only have chromosomes inside them. So there's going to be big circles with chromosomes. I'm not going to draw the nucleus, the ER, the Golgi, and all that stuff. We care about them, but they're not involved necessarily with this whole thing. We really care about what's going on in the nucleus. So if I'm to draw a cell, and inside the nucleus, I'm only going to draw one chromosome to keep it simple. If I were to draw all 46, it would start to get messy. So what you need to know is here's my chromosome, and so this would be my cell, this would be the nucleus, and there's my chromosome. At this stage of the game, this cell is doing its job, it's hanging out in whatever part of your body it's supposed to be at. Say it's a liver cell, and something tells this liver cell it's time for you to divide. You need to split because we need more of you. There may have been an injury. Cells have a certain lifespan and then they die, so they need to split and do their thing. So something has told this cell it's time to divide. It's time to undergo mitosis. So what happens is that this chromosome, and you have times 46 of them, and so this would be our diploid somatic cell, and it has 46 chromosomes. What's going to happen is I need this cell to split into two, and I need 46 chromosomes in every single one of them. And the way the chromosome shape is right now, I can't split that shape and I can't cut that in two. So what has to happen before mitosis happens is that we need to do something called DNA replication. We need to copy our DNA. I need to make an exact copy of all 46 chromosomes. So this little arrow is going to show what happened. We're going to briefly go from here to here, and it's a DNA replication. We'll have a molecular biology unit later that gets into a little bit more details on how DNA replication occurs. So what happens during DNA replication, I'm just going to make it a little bigger so you can see. Here's my nucleus. This chromosome, so if you pretend my arm is a chromosome, this is the top part of the chromosome, this would be the bottom part of the chromosome. This chromosome is going to copy itself into two. And that gives you your common X shape that you see in DNA often. And so if you're ever watching, oh goodness, like Nova or some kind of science things, you'll usually see chromosomes in the shape of an X. That's when they get copied. That's when this DNA that was this chromosome now becomes this chromosome. It's still one chromosome because it's an identical copy of itself. And this is where I don't want the nomenclature of all this stuff to trip you up, because they're the same. This and this are the same, you just have a copy of itself. And you could guess, now that I've made a copy of myself, when I go to split, boom, I could put one copy in one cell and another copy in another cell. When this cell needs to split again, it'll just copy itself again and boom, split again. So that's what DNA replication does. So this chromosome, this guy, which is this guy, actually copies itself to look like that. So now I have 46 of these. It's still a diploid cell. It hasn't quadrupled itself or anything like that. The reason is because that is one chromosome, and this one chromosome has one chromatid, one arm. This is also one chromosome, But instead of having just one arm, we now say it has sister chromatids, 2 chromatids. So it has now 2 chromatids, and we call those sister chromatids. And it's still diploid. But now it's ready to undergo mitosis, because mitosis is about splitting the nucleus, and then cytokinesis comes and splits the rest of the cell. So now when this cell does mitosis, we're going to split this cell in half. Here's my cell, here's the nucleus. Sorry, it got a little smaller. But this chromatid is going to go into here. And this chromatid, here's my nucleus, is going to go into here. And I'm still going to have 46 times 46. So now I have 46 chromosomes, and I'm back to 1 chromatid again. So my sister chromatids split to create identical daughter cells. So these are identical daughter cells. This process right here, from here to here, that's mitosis. And the arrows that you see here over time, we're going to get into the nitty-gritty of that. That's mitosis. But they're identical daughter cells. They're diploid. I want to get all this language common to you. They're diploid because you have 46. You have 2 copies of 23. We didn't mess that up at all. This guy looks exactly like that guy. This one looks exactly like that one. The only thing that had to happen is I had to copy everything before I distributed it. So if I were to, you know, the notes I'm giving you for biology, if someone wanted my notes, like for me to give them my notes, I wouldn't give them my originals. I would make a copy and then give it to them. Or say I had 2 students that wanted my notes. I would make 2 copies of my notes and give it to them. I wouldn't, you know, just get rid of them. So when the cell splits, it has to copy itself so everybody gets everything it needs. So here's where a few more definitions are going to come through. This is going to be number 4 on your list. So number 1 was chromosome, 2 was chromatin, 3 was chromatid, the arms. Now number 4, I'll put it right here, is called centromere. So the centromere is this region right here. It's the region where the 2 sister chromatids meet, right there. The region where 2 sister chromatids meet in a chromosome. So it's that inside spot right here. So if this is 1 chromosome and I copied myself, pretty much where the heels of my hand are, that would be the centromere, that inside point right there. All right, and the last definition we're going to do before we get into how this whole thing works is number 5. This is going to be kinetochore. So it's an interesting spelling, K-I-N, kinetochore. So the kinetochore, since this is going to be our starting point for mitosis, we have this whole process here, that's molecular biology. We're going to get to that later. This is what we care about in this section here. We're going to start from the bottom. So once the DNA gets copied, this is where we're starting. What's happening with this X-shaped chromosome? So here's one copy, here's the other copy, and inside is your centromere. There's this region on the outside of the X, this spot right here. So if this is my X, I'm looking at this spot right here on there. This is the kinetochore region. This is a very important region because when we go to take these 2 chromosomes apart and put them into 2 new cells, what's going to happen is these proteins, some microtubules, if you remember from our cytoskeleton back in the cell day, which was just recently. So microtubules are going to grab into the kinetochore and actually help pull these two chromosomes apart. So the kinetochore is a region on the outside of the chromosomes where microtubules bind to help separate the chromosomes. So a region outside the sister chromatids. So the centromere is on the inside, kinetochore is on the outside. Where microtubules bind to help separate the sister chromatids. So that's why I needed to draw this before I explain these two terms so you can see what I was talking about. All right, now let's get into the cell cycle, because the cell cycle is when we start to learn about mitosis. So this is going to be big Roman numeral 2. Here we go. Oops. Okay, Roman numeral 2, the cell cycle. So the cell cycle is what another way of thinking it would be the life cycle of a cell. So if you're looking at a cell, and if you were to put its lifetime into a pie chart, what you would see are these different pieces of the pie, if you will. So in each phase is labeled something differently. So here you have G1, and I'm going to put one going out here to be G0, and it could always come back out. We're going to talk about these. You have S phase, you have G2, and you have M phase. Let's talk about these really quickly. So G1, G0 as well, G1, S, and G2, these three phases make up the bulk of where your cell is living right now. If you were to think of your whole entire lifespan, the majority of a cell's life is spent in G1, S, and G2. For more realistic terms, G1 is where it's usually hanging out, and it spits a cell out and goes into G0. This is where we call it quiescent, but it's not just doing nothing. G0 is where the cell is actually doing its job. So the majority of your cells are actually hanging out in G0. Your neurons that are all up in your brain, your spinal cord, they don't divide very well. They don't divide fast, they don't divide often. Kind of what you get is what you get to a certain degree past development. They can regenerate slowly, depending on the neuron. Think of your bone. Your bone grows during development and during puberty, but unless you get a damage or a break or something like that, your bone cells typically don't divide very often. So those cells that are doing their job, they're just working away, doing what they got to do, they spend a lot of their time in G0. This is just a happy working cell. G1 is also a happy working cell. And the G stand for growth or gap. Gap phase, growth phase, depends on what the cell is thinking of doing. And so G1 cells, they're also doing their job, but they're ready to understand that at any moment in time, I'm going to be called to divide. So a G1 cell would have a chromosome shape of that. I'm not going to draw the nucleus anymore, just a circle with a chromosome in there. So it would have a chromosome shape of that. They would go through their lifespan, they may hang out in G0, then they might get called back in, and they're going to hang out, and then eventually around here sometime they're going to be told, you got to get ready to divide. So when that happens, then they go into S phase. S stands for synthesis phase. We're going to write all this out in a minute too, but I need to tell it to you. So synthesis phase is when you are synthesizing DNA. You're making a copy of your DNA. So this goes back to that last picture we had up on the board. That's when this cell becomes this cell. it copies its DNA. And now it's realizing, okay, game on, we're gonna divide eventually. And so S phase is over. That's the whole point of S phase, is just to copy your DNA. Your cell functions kind of shut down for a moment so your DNA could get copied, and try not to make any mistakes. And once that's done, then you go back to working again. G2, the cell still looks like this, but it's doing its job, it's functioning, and it's really, really, really preparing to divide now. So at this point in the game, your organelles are getting bigger, your endoplasmic reticulum is getting bigger, the cell's starting to get a little bit bigger because it knows it has to basically split itself in half. You might be replicating mitochondria, you might be doing a whole bunch of stuff, but it's preparing for mitosis. It can also hang out here for a while and just work. So a lot of cells can just work here for a while, do their job, but as they go through time, they're gonna realize we're gonna get the call to split. And this is when M phase kicks in. This is mitosis. M phase is when you get this cell and you split it into the two cells. So this is mitosis, M phase. And all cellular functions shut down for this point because you're just focusing on splitting at this point. You don't want to mess this up. So you don't want to have to think, oh, if I'm a heart cell, I got to continue doing X, Y, Z. There's other guys around me that are working on it. I could just focus on splitting. And that's what's happening here. And then, you guessed it, you just keep going. This guy enters G1, same with this one. They just keep on going through their life cycle again. Now, depending on the cell, this can be rather fast. If you're looking at a red blood cell or a white blood cell or any kind of, your immune cells are beautiful at this, they have a certain replication time that they will copy and divide, but they'll do that very consistently because they don't have long lifespans. Compare that, and same with the liver cells. Your liver cells are constantly regenerating because their liver is the one that's filtering everything you're putting in. So it's working hard. Those cells need to stay fresh, so we just keep dividing and making new ones, letting old ones go. Compare that to your bone cells, your neurons, stuff like that. Your fat cells, they really don't do much. You got what you got. They just get bigger or smaller. They really don't do much. As opposed for dividing. So they have a function. So, those kinds of cells, it just depends on the cell, like what is the life cycle of the cell? It depends on the cell and its function. So that is the whole life cycle of a cell. We are focusing on this guy right here, M phase. That's mitosis. Later on in molecular bio, we're going to focus on the S phase when we talk about how DNA replicates itself and copies itself, But for this section, we're focusing on M phase. The same thing it's the same cycle for gametes as well. A pre-gamete cell goes through all this, and then it enters meiosis instead of mitosis. So we're going to focus on this right now. So let's define what these stages are really quickly. So stages of the life cycle. The first stage is G1, and this can also be called the first gap phase, growth phase. We'll just call this growth or gap 1. It could be first gap, first growth. I've heard many different ways of interpreting this. At this stage of the game, and also for G0, this is when it kind of kicks itself out there. This is when the cell is performing and doing the functions it needs to do, and the cell is just making a ton of protein doing its job. So cell is functioning in whatever role it needs to do, functioning and making protein. Eventually, during G1, it could realize, hey, I never need to divide. It'll kick a cell out into G0, and even then, it could always be called back in. So just know that G0 is like the ultimate workhorse cell. During G1, if a cell gets a signal to say, hey, divide, oftentimes, like if you're thinking of your skin, you're constantly slopping off dead skin. That's the majority of dust that's in the air. It's disgusting, I know, but it's true. But your layers of skin are constantly, you're bringing up new layers of skin every day, so you're making new skin down below. Every day we're replenishing a lot of our skin. So you could imagine those skin cells rarely get kicked out into G0. They're always kind of in this region because they're waiting for the signal, hey, we got to divide. And so S phase, once you get to S phase, you know mitosis is just around the corner. So S phase is synthesis phase. It's synthesis, and this is when DNA replicates and gets copied. So you go from one chromatid for each chromosome to sister chromatid chromosomes. You still have 46. So it's 46, 46, 46, and this is all 46. Nothing, the number hasn't changed here in mitosis. And then for number 3, you got G2. And this would be your growth, your second growth or gap. The second one. And also the cell is functioning, but preparing for mitosis at the same time. And then of course, M phase, and this is where we are going to be for the rest of this. This is mitosis. And so this is mitosis. And when we get to meiosis, it's meiosis. So mitosis is, just think of this as cell division. This is when you actually start dividing the nucleus and dividing the cell. So this is when you get cell division. And that is going to bring us to mitosis. So we're going to start there on our next video. But that is a background so far on everything. What is mitosis? What is meiosis? What's a gamete? What's a somatic? What's a diploid versus haploid? And what are chromosomes and chromatids and all the blah. I want you to get your vocab down before you continue further on with these videos. Feel free to rewind and look and see what's going on. Make sure you get your language down. I'll be good to hopefully remind you as we go through. But this is a whole new set of vocabulary if you've never studied this topic before. So I hope you enjoy it. We're going to get into the nitty-gritty of mitosis coming up next. See you soon. Thanks.